After learning about DNA, have you ever wondered how can a DNA actually result in a trait? Let's take an example, like your hair color. Your DNA has the genetic information that codes for the color of your hair. Your hair color is based on the pigment that is in your hair. But in order to have that pigment, you have genes, which are portions of the DNA that can code for proteins that help make that pigment. Each of us have around 30,000 genes, each of which codes for a certain protein that dictates all of your traits. So what we're going to talk about is how your DNA can lead to the making of a protein to express genetic traits, a process also known as gene expression. Let's have more about gene expression later in this video. So back to your DNA and its essential role to all of this. All of your cells have DNA, well, with a few exceptions, and that DNA is in the nucleus. Although thousands of genes in human DNA codes for proteins, large stretches of your DNA are transcribed but do not code for proteins. These regions are known as introns, and they make up about 95% of the human genome. We also have regions in your DNA that makes up genes that are not activated. But in this video, we're going to talk about genes that are coding for action proteins. So how are we going to get the information from these genes out of the nucleus so that the cells can start producing the proteins that it needs to make? Let us introduce to you to the amazing work of RNA. Remember the process of gene expression which we mentioned earlier? Well, gene expression is essentially consists of two important steps. The first step is called transcription, which is the process of making messenger RNA. The second step is called translation, which is the process of making proteins. The transcription is when we are going to transcribe the DNA into a message. In your cell, the DNA is in the nucleus, so therefore, we're doing transcription into the nucleus. In the process of transcription, the coded information in the DNA is copied to make mRNA. The M in mRNA stands for messenger. So what's something great about being mRNA? Well, in eukaryotes, you can get out of the nucleus. The messenger RNA can go out of the nucleus into the cytoplasm where it is going to attach to a ribosome. The ribosome is going to make or synthesize proteins in the next step called translation. In this video, we will focus on the process of transcription. So the process of transcription consists of two broad steps. First, the enzyme called RNA polymerase II forms a pre-messenger RNA molecule. This happens in the nucleus by using the base pairs complementary to the base sequence of the DNA template strand. In the second part, the pre-mRNA is edited by more enzymes to produce the desired mRNA, which is the final product of the transcription process. So let's look at the first section of this process, which is the formation of the pre-messenger RNA and is essentially taking place in three steps: initiation, elongation, and termination. The enzyme required for the making of pre-mRNA is the RNA polymerase II. RNA polymerase II binds to the promoter region of the DNA. Suppose that this is your gene that needs to be transcribed. At the beginning of the gene, there's a sequence of base pairs known as the promoter region. The promoter region serves as a guide that tells the RNA polymerase II where to attach. Once the RNA polymerase II binds to the promoter region, it separates these two strands of DNA, providing the single-stranded template needed for transcription. Only one of these strands is used in the process of transcription. This is known as the template strand. This strand always runs from the 3' to 5' direction. RNA polymerase II builds the new strand from the 5' to 3' direction, adding new nucleotides to the 3' end. So to speak, the template strand and the mRNA are anti-parallel with each other. The RNA polymerase II, along with other proteins known as the transcription factors, collectively known as general transcription factors, are required for the initiation of transcription. Let's see how transcription factors are designated. The initial two letters are written as TF, which stands for transcription factors. The 2 indicates that it's a transcription factor for RNA polymerase II. The letters next to 2 can be A, B, D, E, F, and H, depending on the function of the transcription factor. Because there are so many transcription factors involved in the eukaryotic transcription, it would be easier to memorize them with the help of the following mnemonics. For example, the mnemonics for TF2A is apple, TF2B is bird, TF2D is dog, TF2E is elephant, TF2F is fish, and TF2H is horse. Let's see the functions for each of them. In the first step of transcription initiation, the transcription factor TF2D binds to the TATA element in the promoter region of the DNA. TF2D has a protein called TBP which binds to the TATA sequence. TBP stands for TATA binding protein. TF2A helps in stabilizing the binding of TF2D with the promoter. TF2B interacts with TBP and the promoter region downstream to the TATA sequence. TF2B helps in the recruitment of RNA polymerase II on the promoter. Now, the RNA polymerase cannot bind to the promoter on its own. A transcription factor, TF2F, helps the RNA polymerase II to bind to the promoter. TF2F interacts with TBP and TF2B while recruiting the RNA polymerase II. TF2F also prevents RNA polymerase II to contact DNA outside the promoter. Next, a transcription factor called TF2E binds to the pre-initiation complex. TF2E helps in the binding of other transcription factor called TF2H. TF2H is a large complex with a total of 9 subunits. Out of 9, 2 subunits help in the ATPase activity. Using energy from ATP, it acts as a helicase and melts the promoter, and this finally causes transition from pre-initiation complex to open complex. The remaining 7 subunits of TF2H has a kinase activity. This kinase activity phosphorylates the C-terminal domain of the RNA polymerase II, and this is where the transcription elongation begins. The transcription factors that helps in the elongation are called elongation factors. There are 2 elongation factors, TFEB and TF2S. TFEB is recruited to RNA polymerase II by transcription activators. TFEB is a kinase protein and phosphorylates serine residues in the C-terminal domain of the RNA polymerase II. This process of phosphorylation stimulates elongation. The other transcription factor involved in elongation is TF2S. Now, the speed of transcription is not the same at all DNA sequences. Some are slow, some are fast. TF2S helps speed up the rate of transcription at the regions where the rate of transcription is very slow. It also doesn't allow the RNA polymerase II to pause, and it pushes it to move on. As the RNA polymerase II proceeds to the process of elongation, the copying of the bases of the DNA template strand to form the new complementary RNA strand begins. The RNA polymerase uses nucleotides according to the base pair complementary rule, but in this case, adenine always pairs up with uracil instead of thymine. So it means that there are no thymines in the base pair sequence of mRNA. And this is how the enzyme RNA polymerase II adds new base pairs on the 3' end of the growing mRNA chain. And this completes the process of elongation. The last step to DNA transcription is termination. And just like the starting sequence of the gene known as the promoter region, which signals the enzyme where to start, we also have sequence of the gene that basically signals the enzyme where to stop. This sequence is known as terminator region. As the RNA polymerase II reaches the terminal region of the gene, the C-terminal domain of the RNA polymerase II interacts with two proteins, CSTF and CPSF. CSTF stands for cleavage stimulating factor, and CPSF stands for cleavage and polyadenylation specificity factor. When the end of the gene is transcribed into mRNA, these proteins are recruited to the mRNA by the C-terminal domain of the RNA polymerase II. The CSTF cleaves the mRNA. Once the mRNA is cleaved, CSTF dissociates. The CPSF then recruits poly-A polymerase, which adds about 200 adenine residues at the 3' end, giving life to the poly-A tail. Once the poly-A tail is formed, the poly-A binding protein binds to the poly-A tail, and during this step, the CPSF is released from the mRNA. The poly-A binding protein prevents the degradation of the poly-A tail. Finally, the newly formed pre-mRNA is released from the transcription complex. So this is the first section of this process in which the DNA is used to produce the pre-messenger RNA. The prefix is used here to show that mRNA is not yet ready to leave the nucleus and start the process of translation. The next section of this process is how this pre-mRNA is gonna be polished to a desirable mRNA molecule. Imagine that you want to publish a new book and you have already printed all the pages. Now, the book is not yet ready because a new book needs covers, both front and back. But before putting it out in the market, it is important to check for any blank or wrong pages that may have been inserted. The two processes essentially represent how the pre-mRNA is processed. The first step involves the addition of a cap and a tail to the ends of the pre-mRNA molecule. These cap and tail are short amino acid sequences that protect the mRNA molecule. The second step of the process involves the removal of any unuseful or junk sequences that do not code for proteins, and these are known as introns. This process is known as RNA splicing, in which the protein RNA complex known as spliceosome removes these non-coding parts. And that completes the process of DNA transcription. The messenger RNA, which is the final product of DNA transcription, is now ready to leave the nucleus, be released in the cytoplasm, and reach the ribosomes. This will initiate the process of DNA translation, which is the synthesis of proteins, or the final step of gene expression.